Great. Welcome, everybody, to today's seminars in oncology lecture given by Roel Verhaak. I was going to have a lengthy introduction, but given that this is being recorded, maybe I'll cut it short. Roel is a professor at the Jackson Labs and associate director of computational biology there. But really, he's one of us. He did his postdoc in Matthew Meyerson's lab, actually around the time that I was in that lab. And I remember thinking that he was an incredibly effective postdoc. Since then, he's went to Texas and then back to Connecticut. But throughout his career, he's been really an incredibly productive person. He's especially been doing a lot of work in glioma and the genomics of glioma, and then in genomics more generally, including emphases in transcriptomics and alterations in chromosome structure, which he's going to talk about today. He's received numerous awards, and he's just been I think, a great leader in the field, in particular showing strength not just in conducting research on his own, but also in leading large groups where he's had a particular skill at being able to bring people together to do group science. So I'm very happy today to present Roel. I should say before I let him talk that any questions that you have, please enter them into the Q&A thing at the bottom of your screen, and not the chat tab. And then when he finishes his talk, then I'll go through and read through those questions. Any questions that we don't get through by, I guess, 5:05 will be sent to him by email, and so we'll connect everything up. Anyway, thanks for coming, Roel, and excited to hear you talk.
All right, thank you so much, Ramin. I need to find the share screen tab again. There we go. All right. Yeah, so Ramin, thank you so much for inviting me to speak here today. It's not the first time I'm doing these seminars on oncology, and it's always a pleasure to be here. Boy, do I think back fondly about that time at the Broad Institute, sort of late 2000s. And what I particularly remember about Ramin is that he was one of the smartest people in an already very smart lab. And what I didn't realize, maybe even until I finished my postdoc, is that Ramin is also an oncologist. I always took him more for a computational biologist, and then to find out that he's actually also treating patients. I think that's probably a good thing that he's treating patients. I'm not always sure about that, but either way, the love and respect is more than mutual. So with that, my lab has been doing and is doing a lot of work in glioma. However, along the lines of doing that, we came across these extrachromosomal DNA amplifications. And because I think that's a very interesting topic, I'm going to exclusively talk about those today and not so much about our work in glioma. To start off with the disclosure, I'm a co-founder and have stock options in a company called Boundless Bio. It's not publicly traded. It's a startup company, and it's focused on developing therapeutics against ecDNA or extrachromosomal DNA positive cancers. So that's relevant here. So as somebody who studied glioma and glioblastoma a lot, these are different diagrams, pathway diagrams with activated and deactivated pathways that drive glioblastoma in particular. There's a number of pathways listed here, including the receptor tyrosine kinase pathway, the p53 pathway, and the cell cycle RB1 pathway, for example. And you'll find that multiple members in each of those pathways are somatically altered via either homozygous deletion, via copy number gain or amplification, and via mutation or via mutations. What you might notice, and they're highlighted here in the next slide, is that quite a few of the members of these various pathways are activated via genomic amplification by extra copies of the same gene. Now, how do genomic amplifications arise? Well, I guess that we don't really know, but we do know that oncogene amplification can be either a linear duplication, where you've got the same DNA segment repeatedly embedded into the chromosome, But we also know that amplification can happen extrachromosomally as double minutes. That's how they were first described. And when they're double minutes, they're extrachromosomal, and we call them extrachromosomal DNA or ecDNA in short. Now, extrachromosomal DNA in cancer, what is that? Now, first, they are circular DNA elements that are typically 50 kilobases to approximately 5 megabases. Extrachromosomal DNAs, circular DNAs also exist in normal cells, but then they are typically much shorter than this, with an upper limit of about 10 kilobases, and they typically do not contain genes. So when I speak about ecDNA in this in the next hour or so, I'm exclusively referring to circular DNAs that exist in cancer cells. Another characteristics of ecDNA is that they do not contain centromeres because they're just an excised portion of the genome, and you know, it would be pretty random if the centromere was included in that, and that's typically not the case. And a very important feature is that it's first and foremost a mechanism for amplification of oncogenes and oncogenic elements to activate the genes and the gene function, to activate the function of those genes that reside on the ecDNA. Now, extrachromosomal DNA is not new. In fact, it was first described in 1965 in a paper in The Lancet, amongst others, where the authors did a very classical metaphase spread and identified these little, let me get the pointer out, these little speckles that could be easily mistaken for dust on the slide. And they identified these speckles typically occurring in pairs, and hence the term minute bodies or double minutes. A few years later, for example, in this paper, these double minutes were related to homozygously staining regions. So it was recognized that you could have a homozygously staining region, a linear duplication of a DNA segment. And the recognition here was that ecDNAs contained the same sequence as these homozygously staining regions. So this was the first indication that ecDNA was in fact a mechanism for amplification. And then just a few years later, a discovery by Fred Alt, but also in parallel by Andy Shimke at Stanford and Dan Haber, I think at that time in Boston too, I recognized that oncogene amplifications mapped to, that it's actually a mechanism for oncogene amplification. So in this particular paper, the gene MYC was identified to be present on the ecDNA elements. So early 80s, we actually already knew that double minutes were a mechanism for oncogene amplification. However, in the next few decades, there's surprisingly little literature on ecDNAs, and perhaps that's in part because there wasn't really an understanding that this is a frequent mechanism of amplification in cancer. Now, why is my lab working on ecDNA? In this paper from 2013, which is actually the very first paper from my own laboratory, we identified breakpoints in the glioblastoma genome. We took all the TCGA copy number profiles and just simply looked, where do breakpoints, where do DNA segments typically, how are those distributed along the genome? And are there regions where breakpoints frequently intersect with genes? So that was really the starting point of this particular paper. And by doing so, we, amongst others, mapped or counted the number of breakpoints across regions, and then realized that there's this region on chromosome 12 and particularly Q14 that is quite enriched for the number of breakpoints. And then when we used the integrated genome viewer, another one of these tools developed at the Broad Institute during the time that Ramin and I were there, and that I still use, in fact, I guess all the habits die hard. And when we looked at the copy number profile of glioblastoma tumors in IGV, it's easy to appreciate that there's actually a highly heterogeneous pattern here, with many of these genomes containing a intermittent amplification deletion pattern. That's quite particular, let's say. However, the realization that this had anything to do with extrachromosomal DNA is because one of the genes in this region is the MDM2 gene, and MDM2 stands for murine double minute. a term that I probably hadn't heard up until then. But when I started reading about it, I recognized double-blind newts to mean extrachromosomal DNA. And this was the first suggestion that extrachromosomal DNA could have something to do with glioblastoma. Around the same time, we started a collaboration with Henry Ford Hospital with an oncologist there named Tom Mickelson and Anna De Carvalho, who was at that time a research scientist in Tom's group. And our collaboration was relatively straightforward. When you take glioblastoma tumors out of a patient, you culture these cells as neurospheres, and you take these neurospheres and you put them into a mouse. What's the genomic drift that occurs in that process? And what does that mean for evaluating drug responses in model systems? In order to answer that question, Anna and Tom collected glioblastoma samples from 13 patients. Anna then created 13 neurosphere cultures, and then she put those intracranially into a mouse, into a not-skid mouse. Had the tumors grow for a while, and then collected specimens and sent those together with the original patient specimen to us at that time at MD Anderson. And then with Hoon Kim, who was in my lab at that time, we performed whole exome sequencing, whole genome sequencing, RNA sequencing, and some long-read sequencing on each of those specimens. Now we had 13 GBM specimens, which means 13 neurospheres, and we did the xenografts and triplicates. So in total, we had 65 samples that we sequenced. What of course is always the first step in any traditional genomic paper is that you create a landscape figure, and this is the landscape figure from that particular project. Here the columns reflect each of the 13 patients, and within each of the columns you'll see the original tumor sample, the neurosphere derived from the tumor sample, and the xenografts derived from the neurospheres. On the y-axis, you see the genes that are typically altered somatically in glioblastoma, including, for example, EGFR and CDKN2A, also known as P16. And you see these are pretty typical patterns. There's about 4 tumors, maybe 5 tumors with a focal EGFR amplification. And what you maybe also appreciate is that the EGFR amplification is propagated from the patient sample to the neurosphere and to the xenograft. So in general, we understood the genomic drift to be relatively limited. However, as a side effect of this study, we realized that we could use the whole genome sequencing data that we had collected to predict the presence of extrachromosomal DNA amplifications. And that is because if you look at one particular example, this is a focal MYC amplification that occurred in a neurosphere from this particular patient. You can see that the focal amplification consists of 2 segments surrounding the MYC gene, partially overlapping the MYC gene. And you might also appreciate that there's sequencing reads that can connect each of the 2, that can connect both sides of both segments, and that you can in fact create a circular path. For example, starting here, you can create a path that tiles along the way of the first amplification, the first segment. You can jump to the next segment, tile that path, and then jump back to the starting point. So with that circular graph, you've got an indication that this is a circular DNA and therefore a extrachromosomal DNA. So realizing that the data had that potential, we tried to do that analysis that I just described. And to validate our ecDNA predictions, we performed a FISH analysis. We did that on the tumor sample and an interface FISH, and you can see that there's many specks in red, so that meaning that there's lots of MYC amplification. And when you look at the metaphase FISH here in the middle, you can hopefully see that the red speckles are in between the chromosomes in blue, and therefore this is an extrachromosomal MYC amplification, as we had predicted. We then scaled up that same analysis, so we did MYC in this particular case, but we also did other genes and other samples, and EGFR, for example, was amplified in more than one case. In total, we did 65 FISH experiments, and comprehensively showed that the whole genome sequencing data was very useful in predicting the presence of an ecDNA. Now, why was that useful in the context of this particular study? Well, we had two patient samples with a focal MET amplification, which is otherwise pretty rare rarely amplified in glioblastomas. What, however, was very interesting to us is that this focal MET amplification disappeared when the cells were cultured. However, when the cultured cells were put into the brain of a mouse, the same focal amplification reappeared. And we know it's the same focal amplification because the breakpoints are identical here and here, and then separately here and here. So this is a very good example of a clonal selection where the majority of cells in the patient tumor contained a MET amplification. However, when the cells were cultured, this turned into a minority of cells and the MET amplification was barely detectable. Put back into a mouse, apparently the MET amplification had a competitive advantage. Therefore, the MET amplification reappeared. And we showed that this is an extrachromosomal amplification here in the fish assay. Then when we compared the presence of a MET amplification in this particular tumor sample to the presence of particular point mutations, somatic single nucleotide variants, we had expected to see a similar strong clonal selection pattern where particular mutations would disappear in the neurosphere and reappear in the xenografts, exactly tracking with that particular clone carrying the MET amplification. I hope you appreciate that that is not the case at all. In fact, the neurosphere has acquired a bunch of new mutations, and on top of that, the xenografts continue to acquire additional events. And there's no single mutation that actually disappears in between. So we published this work in 2018 and along the lines of this particular model where we have a patient tumor that contains different clones when you define clones on the basis of mutations, of groups of clusters of mutations. However, there's another layer here, which is the presence of an extrachromosomal DNA element. When we cultured the cells from this 3035 patient, the clones continue to exist, maybe change a little bit, and in different clones, there was a differential presence of ecDNA. And again, when cultured into, xenografted into a mouse, the ecDNA cells sort of within each of the clones took over again, meaning that it's actually the extrachromosomal DNA that's driving the evolution of this particular tumor. And that's really what grasped at least my interest in studying ecDNA, the very strong driving force in tumor evolution. What I also thought was very interesting is the rapid pace at which a tumor could become a majority amplified versus a majority non-amplified. Now, if you look at a chromosomal alteration, let's say a point mutation in P53, that point mutation during mitosis will be replicated, and the two daughter cells that come out of mitosis will both carry that same P53 mutation because it was replicated. However, when you look at extrachromosomal alterations and the way those replicate, we don't fully understand how that happens. We know that in general, the extrachromosomal DNA does get replicated, and we assume that the subsequent segregation of ecDNAs happens more or less randomly. And that's because we know that the ecDNA elements do not contain centromeres. Therefore, there's nothing for the spindle complex to attach to. There's nothing that pulls them apart evenly. And therefore, we speculate that extrachromosomal DNA can accumulate pretty quickly. Because if it's the cell that has a lot of ecDNA that has the highest competitive advantage, that cell can, first of all, arise in just a few cell cycles. This is just an example experiment drawn up by my colleague at UCSD, Vineet Bhavna. And if this cell now has the highest proliferative advantage, this cell is going to dominate the tumor moving forward. Now, the concept of uneven segregation was easy to draw out, but actually has not been shown to take place. And based on that, a postdoc in the lab and a fellow of the American Brain Tumor Association, developed a project to visualize ecDNA in live cells. And to do so, she used a method that relies on DETCAS9 to hybridize illuminescing signals to the tail of a guide RNA. That's essentially how it works, which means that we can design guide RNA. And if we can design guide RNAs to exactly map to the breakpoints that's unique to an ecDNA element, we now have a tool, a mechanism to fluorescently tag ecDNAs in live cells. So there's two important considerations here. First, we have to find ecDNA specific breakpoints and map them out precisely. So we do that using whole genome sequencing followed by Sanger sequencing. And of course, we need to figure out using FISH assays whether these breakpoints are truly ecDNA specific, yes or no. Finally, and I'll come back to this a little later, because we are using a single guide, the amount of signal is somewhat limited, and there's a little bit of noise because the single guides are more likely to hybridize somewhat randomly. At the same time, we do think we have overcome these challenges and actually find enough signal to do this. So first of all, as I just said, we use whole genome sequencing of some of our cell line models, followed by Sanger sequencing to fine map ecDNA specific breakpoints and then design guide RNAs that precisely align to the breakpoints. This is just a PCR plot to show that in PC3 cells, the breakpoints do not exist, and in our HF3016, which is a GBM model, we see a very clear signal for these four particular breakpoints. And then here's the breakpoint FISH, showing that this co-localizes with breakpoint-specific guide RNAs. Excuse me. Again, demonstrating, validating that the guide RNA approach works. And this is these are the co-localized CRISPR spots with breakpoint FISH signals, just again to validate that the method works. But then here is where I think is the money experiment, which is we took one of our cell line models, used our EC tag system, which is how we started calling it, to visualize ecDNA elements over a period of time, and in particular in cells undergoing mitosis. Now, this is a representative time course, and you might think like, okay, well, I'm not really seeing any signals here. And I would agree with you. If you look very closely to your screen, I think this is maybe the first time point where you start seeing some signal. Now, the cell is starting to undergo mitosis, and as it does that, it condenses its DNA. And we think that because of the highly condensed nature of the DNA, we're actually having a problem hybridizing our guides to the ecDNA breakpoints. So the earliest that we can start seeing some signal is at maybe 8 hours post start. However, at the end of our experiment, we do clearly find that the two offspring cells contain a different load, a different number of ecDNA elements. So this is a pretty strong indication that that uneven segregation truly is happening. Of course, when we submitted this for review, the reviewers were saying, okay, well, that's fine, but we really need to see this throughout the entire process. So the top experiments were done using a transient system where we transfect each of the portions of our EC tag system separately. And basically we lose the signal when the guides start to leave the cell. We now also have a stable cell line, but the stable cell line, so basically there we have each of the parts of the EC tag system integrated into the genome, but the signal is not as strong. So those are some of the drawbacks that we're facing right now. However, when we use our stably transfected cell line model, we have a pre-mitosis signal, and the ecDNAs are highlighted here in the red dots. This is done automated, so we actually didn't touch it's not like we're just randomly adding some red squares, some red diamonds here. Again, we see that the signal tends to disappear during mitosis, but we now can at least visualize pre and post mitosis and see that there's clearly a very different number of ecDNA signals post-mitosis in between the two cells post-mitosis, and that the total number roughly approximates what is pre-mitosis. So again, we think that we're showing for the first time perhaps directly that ecDNA segregates unevenly. So these are just representative cells that I've shown you so far. Of course, we've done this many times by now, and the corresponding manuscript can be found on bioRxiv. when we apply the same EC tag system to a control loci, namely chromosome 7 and MUC4, a gene on chromosome 3, we see that the number of signals between daughter cells has a correlation of 1 or near 1 for the 2 control loci, but a very low correlation which is not significant for the ecDNA breakpoints. And that's exactly what you would expect if a ecDNA would be segregating unevenly. So that explains some of the potent nature, if you will, of ecDNAs to drive tumor evolution. So older cytogenetics-based studies suggested that extrachromosomal DNA is in fact a rare event in cancer. So this paper is not even that old. I think it's 2011. And here the authors used cytogenetics to predict the overall frequency of double minutes to be 1.4% amongst a stunning number of 54,000 samples. So that's a very high number of tumors. And 1%, it's not nothing in the grand scheme of things, but it's also not a lot. Now, as we had found earlier in our study of glioblastoma models, we can use computational analysis of whole genome sequencing data to detect circular elements. Our collaborator, Vineet Bhavna at UCSD, a professor of computer science there, took this one step further and designed the Amplicon Architect pipeline, which basically predicts ecDNAs by starting with a seed, and the seeds are based on all loci with more than 4 copies along the genome. And random seeds are picked within each of those segments, and then the method tries to create a circular path, build out a circular path from the seed. And that's in short how Amplicon Architect works. We applied that method working very closely with Vineet on lots of whole genome sequencing data, and then post-processed all the predictions into basically to assign tumor samples into 4 categories. The first category consists of tumor samples that contain linear amplifications only. And by linear, I mean relatively straightforward duplications of a particular genomic region along the genome. The second category consisted of tumors containing heavily rearranged focal amplifications. And heavily arranged could mean that there's multiple segments, either intra or intrachromosomal, and perhaps also with different configurations in terms of heptatyl structure. Then there are typical breakage fusion bridge patterns that you can identify through the whole genome sequencing data analysis. And then finally, a category of tumors that contains circular DNAs. Now, I fully believe that whole genome sequencing is a great tool to detect ecDNA, but I also think that it's not a very sensitive tool, and I'll tell you why. First, if any part of the circle is not covered by sequencing, which, and sequencing is to a degree a random event, if you cannot create that full circular path, the amplification is going to be predicted to be heavily rearranged or to be linear. This is particularly an issue when you're analyzing DNA that is somewhat degraded, for example, DNA from FFPE, or when you're analyzing sequencing data that has coverage that's suboptimal, for example, in relationship to the amount of tumor cellularity, the purity of the tumor. If you have a tumor that is relatively low purity, say 50% tumor cells, and the coverage is 30x, it may be more challenging to identify circular DNAs. So with that in mind, what we found is when we used the Amplicon Architect tool, basically on the Peacock cohort, we analyzed over 3,000 tumor samples. We found that at least one circular DNA was detected in 14% of all tumors. And these are all primary and untreated tumors. Some of the highest categories were glioblastoma, 56%, sarcoma, almost 50%, esophageal cancer, and ovarian cancer. We did not detect any circular DNAs in acute myeloid leukemia. And here I'm coming back to the detection somewhat insensitivity of Amplicon Architect, and it's not the fault of Amplicon Architect, but the underlying data. And that there's many literature reports of double minutes in leukemia. So there's sort of evidence that. And with that constraint in mind, we still detected ecDNA in 14% of all tumors in this, of course, biased cohort. This is not a random draw of cancer across a population. It's the highly skewed TCGA and PCOG cohort. Yet 14% is a lot higher than what has been reported previously. And this study was led by Nam Yen and Hoon Kim in my lab, and Nam was previously in Vineet's lab. We have now repeated a similar analysis, and this is preliminary data that's still very much under our own review. Yet I thought it'd be interesting to share it with you here. We analyzed the fantastic data set from the Hardwick Medical Foundation, and I should make a disclaimer here that I'm a scientific advisory board member of the Hardwick Medical Foundation. Still, even I think if I wasn't a scientific advisory board member, I still would describe this as a fantastic data set because it's basically the TCGA with only whole genome sequencing data at 100x coverage and only metastatic tumors. And when we analyzed so far 3,000 of the Hardwick cohort samples, we detect at least one circular DNA in 35%. I do believe that this is in part because of the much higher coverage that Hardwick has in comparison to the PCOG TCGA cohort. So it's not just that metastatic tumors have a higher frequency of ecDNA. I think it's in part contributed by the technology. Despite that, there are some quite prominent differences. I'm just highlighting a few here. So for example, breast cancer, metastatic breast cancer has a frequency of more than half of tumors containing at least one ecDNA. Colorectal tumors from 5% to 32%, prostate from 2% to 35%. So these are some really significant increases. One characteristic of Hardwick, and I didn't split out the data further than this, but it is that there are local recurrences, lymph node tumors, as well as what I would call true metastatic tumors in other organs than where the tumor was originally diagnosed. So I think there's more mining that we can do here to understand the nuances and to what degree ecDNA is truly driving that metastatic process. Nonetheless, I think we're just starting to understand the frequency and the penetrance of circular DNA in cancer. So when we, and this is not returning to the PCOG TCGA data, so this is in primary cancers only. When we reviewed which oncogenes are most frequently detected on ecDNA, many of those were also reported as frequently amplified in Ramin's classic tumor copy number landscape paper from 2010. For example, we find MDM2, MYC, EGFR, and others. MDM2, of course, is kind of interesting because it's murine double minute 2, and it's one of the genes that by frequency has the highest, or by proportion in terms of circular amplification versus non-circular amplification, has the highest frequency. But also genes like MYC, EGFR, basically every oncogene that is amplified, that is known to be amplified, is sometimes amplified on a circular DNA. We also found that circular DNA gives rise to extra high gene expression. So we compared the expression level of genes on a circular on an amplification, and then grouped them by amplification types, so circular, BFB, rearranged, or linear, and then compared transcription level to copy number level, because we know very much that copy number level is a primary driver of gene expression. And I hope you'll appreciate that despite the copy number correction, circular DNAs expressed at even higher levels than linearly amplified DNAs. And then we also looked at survival, and of course survival here is biased by the fact that ecDNA is frequently detected in poor outcome tumors like glioblastoma and sarcoma. But even when we do a multivariate analysis where we correct for lineage, we still find that circular DNA has a significant association with worse outcomes. We could attribute this, and that's speculation, of course, to the fact that ecDNA helps the cells to rapidly diversify and thus to overcome any barriers that might exist in the microenvironment. And those barriers could be therapeutic, or those barriers could be in nutrients. It gives cells an extra, or tumors an extra line of defense to circumvent anything that's trying to block them from growing quickly. So a very a big attraction for me personally to join the Jackson lab was the presence of Chailin Wei. So Chailin, together with her collaborator Yi-Jin Ran, has in the past developed a method called chia-pet, which I think is kind of funny because her name is Chia-Lin and then the method is called chia-pet. So chia-pet is essentially similar to Hi-C, so it's a method to measure chromatin interactions and chromosomal interactions. Based on our work on ecDNA, it was a small step to then perform chia-pet analysis on samples that or cell line models that we knew were ecDNA positive, to understand what's the 3D genome, what's the chromatin interaction landscape of extrachromosomal DNA in comparison to other loci on the genome. So when we did this experiment, and we did this with Yan Fen in Chailin's lab and with Amit, who's in my lab, we focused on, we actually took an intermediate step, which is to isolate DNA and fragment DNA, cross-linked DNA, but then to pull down DNA with an RNA pull-2 antibody to focus on transcriptionally active DNA interactions. So when we used RNA pull-2 bound chia-pet and evaluated interaction frequencies, and we first looked at a cell line containing a focal amplification of EGFR, and this is highlighting the EGFR locus. You can see that there's this entire hairball of interactions going on at the focally amplified ecDNA locus. The second cell line here is ecDNA negative and is shown to reflect the contrast here. So there's a network of interactions in intra-ecDNA interactions. And the way I picture this in my mind is that you have a nucleus. This nucleus contains many different floating circular DNA elements. And because they're more or less free floating, I don't think they're entirely free floating, but they're in a small nucleus, they have much opportunity to interact with each other, and that's what we're seeing here. So this is one locus in one particular cell line, and here's another locus, the MYC locus in 2354. And again, that's actually the same MYC amplification that I showed earlier, consisting of 2 DNA segments. And you again see this total hairball of ecDNA interactions that we don't find in the cell line that doesn't contain that same ecDNA. So perhaps this is not too surprising, but what was really intriguing to us is that when we mapped or counted the number of chromosome interactions by, I think we used 100 bins of 100 kb in size, and we counted that across the genome. You can see that there's a huge peak here in the number of chromosomal interactions of the locus that contains the ecDNA. In other words, the ecDNA is making connections genome-wide at a very high frequency, and at a much higher frequency than any other locus across the genome. And keep in mind that this is RNA pull-2 bound ecDNA. We then performed ChIP-seq and mapped and grouped the genome into different categories. The first category, and this is in 5 cell lines in total, with 4 cell lines being ecDNA positive and 335 is a ecDNA negative glioblastoma line. And we grouped the genomes of these 5 cell lines into 3 categories. The first category is H3K27 acetylation of loci that have ecDNA interactions. In other words, in red, you only see the, in the A category, the A's are here, and the A category is just the ecDNA loci in these 4 neurospheres. And as you can see, in the ecDNAs contain very much open chromatin as marked by H3K27 acetylation. We then have a second category, which is loci where chromosomal loci where ecDNA is predicted to make a connection on the basis of our chia-pet data. And then the third category is everything else. And I hope you appreciate that the chromosomal loci where ecDNA is predicted to make a connection very much has also open chromatin. then finally, when we reviewed the gene expression level of genes very close to the B category here, we find that genes that are predicted to be in contact with ecDNA have significantly higher levels of gene expression than genes that are not in contact with ecDNA. So all of this to suggest that ecDNA is actually driving gene expression of chromosomal genes. It took us a long time to get this study published. And I'm happy to say that it got published earlier this year. It made its journey through various journals. And the number one critique that reviewers had was, okay, you can convince us that ecDNA is having this effect, but can you show us that it matters? And of course, we argued, well, that's for our next study, not for this study. And I'm glad that we finally found an editor willing to agree with us on this. And I do want to highlight that it is our next, very much our next step, is to understand, does that effect of driving transcriptional activity really have an impact? Of course, we're using single cell techniques to understand this better. So then, in my final few slides, I want to show you some, I think, very interesting data that suggests that ecDNA tend to cluster, and it builds on the back of this transcriptional activity effect that ecDNA tends to have. So we used our EC-tag method again to create a time series. Actually, we just noticed this pattern when looking at lots and lots of images. And by we, I really mean in he. It's reflected here. You can see this particular nucleus that has ecDNA as marked in the green. And as time progresses, these ecDNAs tend to sort of converge. Now, this is just one example. It doesn't really mean much if you think about it. But then when you look at lots and lots of images, we notice that this is actually a real pattern that perhaps is not as random as we initially thought it was. So we see that ecDNA tends to cluster. In fact, when you count over time lots of cells, we find that about half the cells marked by our EC-tag method contains clustered ecDNAs. And in contrast, none of the cells that we marked with chromosome 7 or MERK4 controls ever showed this ecDNA clustering. Now, that's one thing. The next thing is that we looked at RNA pol 2 again and found that these ecDNA clusters had a preferential convergence with RNA pol 2. So it's not just that ecDNAs tend to form hubs or clusters. It's that those hubs converge with RNA pol 2, suggesting that they have a function which is to drive gene expression of ecDNA cargo genes. Again, this is the reflective example, and this is the data compared between the ecDNA breakpoints and the two controls and co-localization with RNA pol 2. And then finally, we looked at whether we used RNA fish, immunofish, to see if clustering actually makes a difference in the expression levels of the cargo gene. We focused on a EGFR ecDNA in this example and find that clustering ecDNAs that are clustered produce much more EGFR than ecDNAs that are not clustered. And these results are also described in our bioRxiv paper. So I think these are some interesting observations with respect to the effect of ecDNAs on gene expression. But keep in mind that in my view, we're only really scratching the surface of ecDNA. I think we're at the start of maybe a new direction in cancer research. And of course, ultimately, we hope that studying ecDNA will give opportunity for development of new therapies to slow growth of cancer. So I'll close by summarizing our findings. Intratumoral ecDNA heterogeneity is driven by uneven segregation. I think we've speculated that, we've now shown it. And as a result of that, ecDNA elements can rapidly accumulate, and which then enables a competitive advantage for cells loaded with ecDNA-driven oncogenic signals. We find that extrachromosomal DNA amplifications are commonly found in cancer, much more commonly than we previously thought, and they associate with poor cancer outcomes, perhaps because of that rapid heterogeneity effect. And glioblastoma is probably the most frequently associated with ecDNA, which may be one reason why we found it relatively early on in our studies. And another, or a leader in the budding field of ecDNA is Paul Michel, who is a neuropathologist by training. And I think it's no coincidence that it's the glio people that maybe are jumping on this relatively early on. And then finally, I think we've shown that extrachromosomal DNA can function as a mobile enhancer element, which then can cause what we've called synthetic aneuploidy, because it increases gene expression not by chromosome, but genome-wide. And we're still working to understand precisely what the impact of that is and how to potentially, and whether blocking this synthetic aneuploidy makes a difference in cell growth. So I'll close by thanking my collaborators, Paul Michel and Vineet Bathma. So Paul is also the leader of the Grand Challenge application that we're currently preparing, as ecDNA was one of the 10 or so topics that could be selected for Grand Challenge applications. My internal collaborators, Chai Lin, as well as Albert Cheng, who previously developed the Casilio method, and we based our ECTag method on discoveries he has made for Casilio. And then finally, of course, the lab and our funding agencies. Thank you very much for your attention, and I'm happy to take questions.
Great, thanks, Roel. This is a really fantastic and far-ranging bit of work you were describing. We have several questions right now, and they're coming in, I guess, in real time. So I'm going to start by asking the first one, which is, are extrachromosomal DNAs found in similar samples as chromothripsis, chromoplexy, BFB cycles, etc.? Is there an overlap in target genes, samples, and even double positive caused by algorithms? Is it possible that one event that is mistaken for another in whole genome sequencing analyses?
That's a lot of questions in one question.
Right.
I'll try to address all of them, but I probably am going to forget. So first of all, in our pan-cancer analysis that we reported last year, we in parallel and independently identified BFBs using a separate algorithm. And that's why in our classification, we first took out all amplifications that were classified as BFBs. So I think we have presuming that the algorithm to detect BFBs was accurate, we have taken those out of the equation before we classified any amplicon as circular.
But actually, sorry, just to stop you there, but you had described how ecDNAs could be mistaken for non-circular. So is it possible that some of those look like BFBs, for example?
Yes, but that's certainly possible. I would say that because we first ran a BFB detection algorithm, that perhaps some circular DNAs were misclassified as BFBs, and therefore our prediction of the circular DNA frequency may have been decreased by a little bit. Again, this is somewhat speculative because we don't really have truth data. In our paper, we do have truth data in that we analyzed a lot of cell line data on which we were then able to do FISH. So I strongly believe that our circular DNA predictions are good. In fact, we had a 90% accuracy in this cell line cohort. Surely it's not perfect, and I do certainly also think that we are not super sensitive, that we are underestimating the true frequency of ecDNA. Now, the chromothripsis question we also tackled by running Peter Park's, I forget the name of the algorithm, but we ran their algorithm to detect chromothripsis, Shatter-seq maybe, and found that approximately a third of the circular amplifications that we had detected converged with a prediction of chromothripsis. Now, chromothripsis is a mechanism for is genome shattering. In itself, chromothripsis doesn't create an amplification. An amplification occurs when the chromothriptic loci is doubled, and then that gives a proliferative advantage. So we tease apart those events. But in about a third of cases, we find that the amplification was caused by chromothripsis, but it also means that 2/3 it was not. So there's some convergence, but certainly not one-on-one. What did I miss?
Yeah, I mean, is it possible that one event is mistaken for another? And I think you've addressed that.
Yeah.
And is there an overlap in target genes?
Yeah, absolutely. I mean, as I mentioned, the genes that were in your 2010 paper are more or less in fact, we used your 2010 paper to look at those are the 24 genes we've listed. And again, your 2013 paper, I think, was actually the direct source for the 24 genes we were showing. So if a gene is amplified, some portion, some fraction of that genes across a cohort will be circularly amplified on ecDNA. So certainly it's very similar.
Great. So the next, I guess, set of questions was, did you see any evidence that these extrachromosomal DNAs get integrated into chromosomes and stabilize? Well, that's the first of two.
Let me start there. Yeah, that's a great question. I mean, Don Cleveland published a paper in Nature at the start of the year where I thought they did really beautiful experiments following the development of a amplification in a HeLa cell line treated with methotrexate, and then propagating that same cell line and doing whole genome sequencing along the way to show the development of the amplification. And they showed that under high constraints, under high methotrexate constraints, you can create an extrachromosomal DNA. They also find that when they reduce that pressure, they can sort of make, quote unquote, the circular DNA reintegrated to the genome. And they even show that they can direct that reintegration through CRISPR by creating DNA breaks. You'll see preferential reintegration where the break was created. Now that's in a cell line model, and how exactly this translates to what happens in patient tumors is less clear. When you do whole genome sequencing, you take a hammer and you slap all the cells in that tumor and you create a flat genome essentially. So I think we would need potentially single cell DNA sequencing to fully tease that apart, and we're not quite there yet.
Great, and then the second question for this person is Xiuan Zhang is, do highly aneuploid tumors have more ecDNA?
So I guess we first need to define what aneuploidy means. And so my interpretation of what aneuploidy means is an aneuploid tumor is a tumor that has lots of chromosome arm or chromosome level amplifications or deletions. And since Ramin and I trained in the same environment, we're probably quite aligned on that definition. And if that's the definition, then there's a little correlation between the presence of ecDNA and aneuploidy. Now, there are certainly tumors that have more focal genomic events than others. And I guess I refer to that as genomic instability, which I think is a little different from aneuploidy. And there's a clear correlation between more unstable tumors and the presence of ecDNA, but not so much on the chromosome arm or chromosome level events level. Yeah.
Great. Chadi Alfaran wrote, thank you for this wonderful talk. What kind of NGS library was used for Amplicon Architect? Whole exome sequencing?
Since you need to capture the entire circular path, it's almost impossible to not do it without whole genome sequencing. So you need whole genome sequencing. With exome sequencing, you have to be extremely lucky to be able to create that full circular path. I suppose it's not impossible. Now there is a method called CircleSeq. It was developed by a group in Copenhagen. Had you known, you could have gone on your trip to visit them. It's a very nice method. Although in my lab, we haven't been able to get it to work yet. That's probably because we haven't tried hard enough, and we're not certainly experts in this area. I know people in Berlin have gotten CircleSeq to work, and I know that because they published a Nature Genetics paper on neuroblastoma where they used CircleSeq. And in fact, we're collaborating with that same group led by Anton Hansen because they also have single cell CircleSeq working, and I'm hoping that we can implement that in the future too.
Great. Great. So I think we don't have any more questions in the Q&A. Oh, well, here, Ming Tang is asking, curious how you target ecDNA for treatment.
Well, so what we are finding is that ecDNA, for example, segregates unevenly. We speculate that the segregation must have some that there's probably an ecDNA specific way of dividing tumor cells, of dividing ecDNAs between tumor cells. We're also speculating that there might be ecDNA specific DNA replication mechanisms. It's all speculation, but we're hoping to show in the future that that's really true. But those would be targeting those mechanisms would be ways to target ecDNA.
I mean, in theory, could you, if you were able to apply CRISPR, obviously in tumors that's hard, but CRISPR to ecDNA, you could kill cells just by the number of cuts you make, and you could presumably target just to the ecDNAs because you have the rearranged loci which are unique to those tumor cells, so you wouldn't be cutting normal cells. But
yeah, it'd be highly individualized because you would have to sequence the tumor and then precisely figure out where the breakpoints are and so forth. So I don't know how scalable that would be, but in principle, I think it's possible. Certainly in cell line models, I think I've seen data to that extent already.
Great. I have a bunch of questions as well as that I left for last. So one is that you showed uneven segregation or distribution of ecDNAs between daughter cells, but have you shown that it's random, or can you?
Well, the one figure that I showed of where we counted the distribution of ecDNA elements between daughter cells is as close as we get to that. So there we took, I think we had about 60 cells that we followed through mitosis, and then we counted in the two daughter cells the number of signals. And we showed that there's essentially no correlation between what's in daughter cell 1 versus what is in daughter cell 2. And that then would imply that it's relatively random.
Okay. Is there do you see anything of it, or have others seen any evidence of ecDNAs clumping at certain part? So, for instance, would they be more likely to interact with each other, have hybridized each other, and therefore be in the same parts of the cell, and maybe then transit to daughter cells more often together? Any evidence of interactions?
Edit in that level of detail just yet. I think we're working up the technology to be able to do that. I'm actually quite interested in these new imaging based screens where we can take super high resolution images of micro tissues. It's a bit thinking far ahead, but we're trying to get these things to work, and then do that in live cells over time, so that you can study the behavior of ecDNA in live cells in a tissue setting. So that's sort of pie in the sky, but when we have that up and running, we can start to do things like this, I believe. And
so the numbers, as they change through the cell cycles, is it that as the cell goes through mitosis, it doubles the number of ecDNAs, and then they split unevenly between the daughters, and then doubles again? Or do you get things like frequently ecDNAs fusing, as well as re-entering the linear genome?
I think it's probably the first of what you said. As during mitosis, the cells double the number of elements, and then they randomly segregate. That seems to be what we have witnessed so far. I think one of the cool things about biology is that probably everything is true in some cells in some setting. So I don't think it's inconceivable that there's alternatives, but in the cells that we have looked at so far, it seems to be the case that they double and then randomly segregate.
And I have a few other questions. So obviously I had a lot of questions from your talk. So do you have any sense of what determines the size of ecDNAs? Are there upper and lower limits?
Yeah, it's probably the penalty that cells pay for maintaining it is probably deciding the upper limit. At the same time, we have seen in, for example, melanomas that the elements can be very large, up to 20 megabases. And we have also found ecDNAs that are very small, like 150 kilobases. Now, what determines that? There seems to be some tumor type specificity there, but why that is, I have no idea.
And you showed that how MDM2 is very frequently in ecDNAs, but less frequently within amplifications within a chromosome. Conversely, MYC, it's more equal maybe. And do you have any sense of why that is? I mean, for example, could it be due to the local regulatory context that if some genes have more frequent, if some genes have more active regulatory elements, then you could just do tandem dupes to amplify them in the regulatory elements. That's fine. Whereas if they don't, they need more complex events, which ecDNAs provide, where you could bring in more distant regulatory elements. Is that the kind of rule that would be applying?
Oh, that's a great hypothesis. I think we should test that, and with the computational data, with the whole genome data, we should actually be able to do that. So it's a great suggestion. I do think there's some some tumor type specificity here, where perhaps some tumor types tend to amplify MDM2 to quite high levels, and therefore it's more likely to become ecDNA. We're also more likely to detect it as ecDNA if their levels are higher, of course, because there's more sequence. So we have to keep that in mind, that although we do this pan-cancer with thousands of tumors, the number of tumors within a cancer is still pretty small, and we still need millions of tumors in the end to fully address these questions.
I can definitely agree with that. We need to sequence more tumors.
And whole genome sequencing, I think the ACR-Gini data set is phenomenal. There's no doubt about it. But we need those types of data sets with whole genome sequencing. And that's why I so much like what the Hardwick is doing, basically performing a TCGA on metastatic tumors. I think that's wonderful. I hope that we can see more initiatives like that, and that they publicly share their data.
And that the data are allowed across country borders, and with the regulations make it reasonable to access those data. Yeah. Great, thanks. I think there are no more questions here, and I don't have any more. So I'd just like to thank you very much for really interesting, exciting talk, and hope to be able to follow this closely in the future, and hopefully have you back again sometime soon.
Yeah, that would be great, and I look forward to maybe catching up at SNO in November or elsewhere. Yeah. And thank you again for the invitation.
Yeah, of course. Bye-bye now.
Bye-bye, everybody.